Great. I think we'll get started. Welcome everybody to the Connect Science seminar series. Just a couple of announcements. First, I want to thank our board and especially Claudia for all the assistance in keeping these meetings going, and remind everyone that to please put your questions for Dr. Elkebetz in the Q&A session, and we'll be able to go through them at the end of the seminar. So with that, it's my distinct honor and pleasure to welcome Dr. Moshe Elkebetz back. Unfortunately, I wish it were in person, and hopefully we'll have that opportunity soon. But Dr. Elkebetz comes to us from Ben Gurion University, where he's an associate professor, and he has had quite a journey in his training that has led him to where he is now. He started off doing his PhD and training with Ronnie Apte, where he made some really important insights into IL-1 beta signaling, and particularly in myeloid-derived suppressor cells and their role in cancer progression. So he already hit the ground running when he came to my lab for his postdoc in 2010, which makes me feel very old now. He has brought his very young family with him at the time, who are now grown. And so he made some really important insights in my lab as well into the tumor microenvironment of breast cancer and bone marrow derived immune and hematopoietic cells that secrete factors to aid cancer progression, particularly looking at the role of granulin in that process. And he then went on to do a second postdoc with Jose Paselga, where he really formed the cornerstone of his career. He was looking at actionable signaling pathways, first of all in breast cancer and then in head and neck cancer, where he's really established his career and is becoming a leader in that field, and has established his lab, his research lab back at Ben Gurion and BGU, in head and neck cancer, where he's published a number of fabulous publications. So we're really excited to have him back and hear about his work. So with that, I will be quiet and let Moshe take the screen.
Thank you very, very much, Sandy. It is always a great pleasure to get an invitation for you, and I will always accept them. So thank you very much all. So let's see. So I'm speaking with you from Beersheba, which is the little city in the corner of the world, somewhere in the Middle East. And this is the university that I belong to. It's a very nice one. You're all welcome to be here. And in my lab, at the Faculty of Health Sciences, we study head and neck cancer. Specifically, we develop and investigate therapies against this disease. And I got very excited from the disease because it's a very heterogeneous disease. There are multiple sites that the malignancies can be developed, like the tongue, oral cavity, nasopharynx, or pharynx. And the disease is also can be developed by either tobacco or alcohol, so meaning that the disease is also very heterogeneous. Patients that diagnosed with the early stage, like stage 1 or stage 2, are very well treated with either surgery or radiation, and the survival is very, very good. But patients that diagnosed in the late phase, particularly in the phase 4B, which they have a systemic disease, they are treated with chemo-targeted therapy or immunotherapy, but the survival is quite poor. So there is a unmet clinical need to develop new treatment for this disease. But we face quite a lot of challenges with the malignancy. In addition to the fact that it's a very heterogeneous disease, in the molecular level, there are very few key oncogene and tumor suppressors genes that are known to drive the malignancies. And you can see, for example, the comparison between HPV positive and HPV negative. There are some key oncogenes like the PIK3CA and the P53, but it seems like it's not like melanoma when BRAF and RAS are important. It seems like there are different oncogenes, and this is become a little bit challenged when we try to develop therapies. In addition to that, even that we give a therapy, and the therapy is potent, we know that in some cases, resistance is developed. And basically, there is a long process of therapy adaptation that eventually leads to the resistant phenotype. And the resistance can come in multiple forms. There is an intrinsic mechanism. It means how the tumor cells rewire the signaling machinery that adapt the therapy and basically escape from the treatment. But also there is the tumor microenvironment that is very heterogeneous by itself that also can support to this phenomenon of drug resistance. So where my lab comes to the field, and basically we are doing multiple step process to try to overcome those challenges. So I won't tell you much today about the preclinical models that we developed, but basically this is one of the forte of our lab that hopefully in next time I will be able to share with you more data. But the major topic for today is the process or the understanding that we provide into the field, into the understanding of the intrinsic and the extrinsic mechanism of resistance. And based with those understanding, we are trying to investigate rational-based drug combinations. So today I will tell you a few stories, at least the two stories, the two topics that I will initially tell you are public. So they're published a few years ago. So I will only summarize the highlights, but then I will give you few stories that they are either recently published or they are in the process of manuscript preparation. So when I established the lab, I did my postdoc in Jose Baselga when I worked on the PI3K. And basically, the PI3K is one of the key oncogene in head and neck. The PIK3CA is the gene that encodes to the P100 and alpha subunit, that when this gene is mutated and a protein is mutated, it can activate downstream independent of the receptors. So when I joined Jose Baselga lab, we were trying to understand what is the role of treatment patients with a PIK3CA mutation with inhibitors, specific inhibitors of the P100 and alpha. And over the years, there were few clinical evidence, including this paper, shows that specifically in head and neck, mutation of patient harboring mutation with the PIK3CA seems to respond quite nicely to the alpha inhibitors. And the project that I initially started in Jose, we tried to understand what are the mechanism of resistance and what are the molecular determinants of sensitivity. And the finding is the following. Patient with the PIK3CA mutation, they seems to activate very much the AKT and the mTOR pathway. However, when you give the treatment with the isoform specific inhibitor of the P100 and alpha, you block the downstream pathway of the AKT and mTOR, and you can induce tumor growth arrest that lasts for a few weeks. However, over time, there is a signal rewiring, and we have found that one of the mechanisms of resistance is the upregulation of a receptor named as AXL that dimerizes with the EGFR, induces a unique phosphorylation on the side that can activate PKC and bypass AKT and activates mTOR downstream. And these findings were validated in patients, and we have been able to show that by combining with the EGFR inhibitor, we've been able to prevent this activation and induce potent therapy combination. So when I established my lab, the first question that we wanted to know is what is the transcription machinery that regulates this upregulation of AXL? So Mai was one of my PhD students, okay, and in her work, she showed that as a response to the treatment, there is an upregulation and activation of the AP1 complex. And once it's active, it can bind on the promoter of AXL and induces AXL overexpression. And when genetically or pharmacologically we inhibited the AP1 complex using either SI for Jun and FOS or JANK inhibition, we have been able to abolish this upregulation of AXL, and by that we enhances the efficacy of the treatment. We further ask whether this is unique, whether the mechanism of resistance in HPV positive are also related to the AXL phenomena. So we have used a different drug. This is a drug by Genentech. It's a beta-sparing inhibitor. And basically we developed the same system. We made an acquired resistant cell line, and we asked what are the differences between the sensitive and the resistant cell lines. And surprisingly, we found that in the HPV positive, the mechanism is not via activation of the receptors or upregulation of the receptor, else it's by upregulation of the insulin growth factor 2 that made an autocrine activation that activates the IGF-1R receptor, and by that activate alternative pathway, the MEK pathway, that enhances the progression of the disease. However, and when we inhibited the IGF-1R using an inhibitor, we have been able to reverse this phenotype. We also wanted to understand what are the intrinsic and extrinsic mechanism to the EGFR inhibitors. And EGFR inhibitors are the only targeted therapy that are approved by the FDA. And basically in head and neck, about 80% of the patients have overexpression of this receptor. And when this receptor is activated, it can induces either the MEK pathway or the PI3K pathway activation. And Ofra, also a master in the lab at the beginning, Ksenia, she was also a master, both of them currently a PhD student. We asked what are the mechanism of resistance. So Ofra was found two exciting findings. The first finding is that many investigators and many papers show that when you inhibit the receptor of EGFR, you have an upregulation of HER2 and HER3. And she was the only one that have been shown that the transcription factor EHF is the transcription factor that drives this upregulation of HER2 and HER3, specifically when you inhibit the EGFR and the MEK pathway. Moreover, we had exceptional responders to Erbitux in head and neck cancer patient, and when we did a sequencing of this patient, we showed that this patient had an amplification of MET. So we saw that there is, in one hand, you can either have an upregulation of receptor of HER2 and HER3, but on the other hand, you have also amplification and overexpression of a different receptor known as MET. And what was nice to understand that it can be it's either HER2, HER3, or MET. And basically in the patient that they have a MET amplification, or there is a overexpression of the ligand of MET, you must inhibit the MET pathway. Otherwise, even a combination of EGFR and HER2 and HER3 is not potent enough. And Ksenia did ask the same question, but in different approach. Here, she took different patient-derived xenographs, and she treated them with Erbitux, and she compared the sensitive and resistant. And by sequencing, she have been able to see that the tumor microenvironment is different between the sensitive and the resistant tumors. And basically, she showed that in the resistant model, we have an accumulation of cancer-associated fibroblasts that are expressing high level of TGF-beta signature. And basically those CAFs seems to secrete many factors, including factors of the HGF that activates MET and therefore can induce resistance. So basically with those models, we have been able to, with those studies, we have been able to understand that MET activation involves both an intrinsic and extrinsic mechanism of resistance to EGFR inhibitor. The next story, which was part of it was published, part of it not yet published, we ask what are the mechanisms of resistance to MEK inhibition. And the reason that we want to target MEK is because we know that multiple receptors multiple RTKs can be expressed. So we know that EGFR can activate MEK, but also other receptors. So it makes sense to try to inhibit downstream of the receptors, and therefore we use MEK. In addition to that, we selected a specific MEK inhibitor that was shown in a phase 2 clinical trial by Ravi from the Dana Farber, and he showed that the trametinib is quite potent in patients, and basically you can see that in most of the patients we have a tumor shrinkage. So again, Avital and Ofra and Manu, we asked what are the intrinsic and the extrinsic mechanism of resistance. So Avital wanted to understand what are the signaling that can be activated following trametinib treatment. So she took multiple cell lines that seems to be sensitive to trametinib, and she showed that there is quite fast upregulation of the phospho-AKT, and I see that the phospho-AKT level, it's not shown here, but you can see it in the next slide when we took the multiple. We did the protein array and we looked at the different receptor. We saw that there is an upregulation of the phosphorylation of the EGFR. And when we prevented the activation of EGFR using erlotinib, we have been able to prevent this upregulation of AKT by inhibiting the EGFR. Obviously, the combination seems to be potent when we inhibited the trametinib and the PI3K, which is activated following trametinib. And you can see here the superior anti-tumor activity compared to the single agent. In mice, it was very potent. The combination we took here, PDX harboring an HRAS mutation, and you can see that the trametinib showed some degree of growth inhibition, but it wasn't sufficient to induce stable disease. And BYL is not potent almost at all in this specific model, but the combination seemed to be very potent. And by immunohistochemistry analysis, you can see that we inhibit the downstream pathway of ERK and mTOR by staining for phospho-ERK and phospho-6. And the combination seems to be a reduced tumor cells proliferation indicated by the Ki-67. So basically, this work, although we have not been able to show what is the transcription factor that induces EGFR, but we did learn that once we treat the cells with trametinib, there is an upregulation of EGFR that activates AKT pathway and induces survival. But when we treated the cells with trametinib and BYL that inhibits the PI3K pathway, we've been able to enhance tumor growth arrest. To understand the mechanism of extrinsic mechanism of resistance, we took advantage of the protocol that it was well known for many years. And basically, we use a carcinogen known as a 4NQ to induce oral carcinogenesis in the mice. And when we expose the mice to the 4NQ, we have been able to see that we have an activation of the MEK, of the pathway of the MAPK pathway using phosphorylation phospho-ERK. So the first hypothesis was whether trametinib treatment can actually prevent the 4NQ-induced head and neck cancer in mice. So we have done the following experiment. We took the wild type mice, we gave them 4NQ for 12 weeks, and then we stopped the treatment with the 4NQ, with the carcinogen, and replace it with a daily treatment with either vehicle or trametinib that inhibits the MAPK pathway. And we have been able to monitor the mice, and we saw that trametinib only delayed the progression of the disease, did not prevent it at all. And you can see here that the differences between the vehicle and the trametinib treated mice was quite remarkable. When we repeated this experiment and we analyzed the tumor around 150 days, we saw that indeed in the vehicle we have massive lesions of the pre-malignant lesion, and some of them are also cancers, while in trametinib treated mice, it seems like there is only a delay of the disease. When we analyze the histology of the tumor, we have been able to see the differences between the vehicle and the trametinib. compare obviously the naive tongue, which is normal epithelium. Here you can see the thickness of the epithelium, but in the vehicle treated group, we had the massive disease that actually penetrated into the muscle of the tongue. And this is the quantification of this penetration invasive thickness. We did a further analysis of those tongue, and we asked what is the presentation of immune cells in the tumors. So we did a very simple staining of CD8, FOXP3 for Tregs, and PD1 as a marker of activation. And we'll be able to see that in the trametinib treated, we have a shift from immunosuppressive environment as shown in the vehicle group. We observe more of activating microenvironment enriched by the CD8 that it seems to have expressed low PD1 and reduction of FOXP3. So obviously those experiments are 300 days that it's impossible to conduct research on them. So we developed cell lines from those mice, and we explored the involvement of the anti-tumor immunity in the efficacy of trametinib with those cell lines. So we collected multiple cell lines. Today in the lab we have more of 20 different cell lines, but in this project it's only 2 cell lines, and this is an example of one of the cell lines, and the cells seems to be very sensitive to trametinib. And to understand whether immunity plays a role in the efficacy of the treatment, we injected these MAPK-driven cancer into NSG or wild-type mice, and then we treat those mice with trametinib. And by by measuring tumor volume, we can estimate the contribution of the tumor microenvironment or the immunity in the efficacy of the treatment. And as you can see here, that the trametinib treatment induces a stable disease in wild-type mice, while in the NSG mice, there was a tumor growth delay, but the tumor still progressed a little bit more. To understand what are the immune cells that play a role, we do the simple flow cytometry analysis, and we saw that after a few days of treatment, we have a massive infiltration of CD8 cells compared to the vehicle treatment. And those CD8 cells seems to be very active because they can proliferate very well. You can see here by the CFSC proliferation assay, as well as by the ability of those cells to express interferon gamma, which indication for the activity. Functionally, we depleted those CD8, and we wanted to understand what is the role of those cells in the tumor. And as you can see here, in the tumors treated with trametinib and depletion of CD8, we have been able to, the tumor cells been able to rescue, to escape from the treatment much to a greater degree compared to the mice that treated with trametinib and IgG, indicating that CD8 does play a role. CD8 cells do play a role in the escape mechanism, or even to say that the potential they can enhance the efficacy of trametinib. We further analyze the tumor at the late stage, and you can see here that you have a tumor stable disease, but the tumor progressed. And when we analyze those CD8 cells, we saw that they are expressing high level of PD1, TIM3, indicating for the exhaustion phenotype. Based on that, we speculated that if we can reverse their activation by supplementation with a PD1, for example, we've been able to induce a greater response in mice, meaning that more mice will respond. So you'll have the better survival, and the duration will be much longer, as speculated by Sharma and Allison. And basically, we followed the same principle. We took mice, we injected them orthotopically either to the lip or in the tongue. And basically, we started treating them with trametinib, and then we supplemented them with the anti-PD1. And as you can see here, that the tumors are quite resistant to the PD1, because then maybe there is a tumor growth delay, but eventually all the mice develop. Similarly, also to trametinib, that you have a stable disease, but then tumor progression. but the combination was extremely potent. And this is an example of one experiment of the 4QL, but across all the experiments that we did with this model, all the mice were survived. And the mice were cured, and I'm saying cured because when we stopped the treatment after 40 days, none of them actually developed cancer. In the 4QT, the scenario is less optimistic. Here we get about 60% of the mice actually cured. And we assume that if we'll give the treatment for longer, we will be able to rescue more. But we, as I said before, we stopped the experiment at the 40 days, and here they progress about 20 days afterwards. This is an example of efficacy of 2 models, but we tested it in 2 additional models, including the mirror cells that they have a HRAS mutated E6 and E7, and this is the model that we developed in the lab. And basically, you can see that the combination is extremely potent in most of the models. Further analysis of the tumor microenvironment of the treatment combination, we saw a massive infiltration of CD8 that express high level of granzyme, and the mice that were cured, they had developed an immune memory, because when we reimplanted tumors into a cured mice, none of those mice developed cancer compared to the naive mice that all of them developed cancer. Furthermore, when we injected the cured mice with a different type of cancer, like melanoma, all of them developed cancer, indicating that we have a specific immune memory, and this treatment combination induces an immune memory, which is crucial for prolonged response. We then wanted to understand whether the timing of treatment is important, because I told you before that there is an adaptation process, and when you supplement PD-1 or you want to induce the anti-tumor immunity with the checkpoint blockers, maybe the timing is crucial. So what we have done, we asked a simple question, whether duration of trametinib and then supplementation of anti-PD-1 can change the efficacy of the treatment combination. And as you can see here, this is a 5 arms experiment, and here in the green experiment, it's like you saw before, that you have 5 days of treatment and then supplementation of anti-PD-1, and you have a tumor elimination. But in the other group that we gave 25 days of treatment and then added PD-1, we had a slight tumor growth rates, but all the tumors eventually developed cancer. So the question is, why does a short treatment with trametinib sensitize the tumors to anti-PD-1 while prolonged treatment with trametinib didn't work? So we did a site of analysis on the tumor microenvironment before treatment, after 7 days of treatment, after 30 days of treatment, and we have been able to profile tumor microenvironment, and this is what you can see. And here is the labeling of the CD8, and you can see that the CD8 in untreated, they express anti-PD-1 indicating for the suppression activity. However, after 7 days of treatment, all these CD8 exhausted cells disappear, and there is accumulation of a different immune CD8 cells. It seems to be the active one that we measured before with the activation of the interferon gamma. However, over time, if you prolong the treatment, you get again reaccumulation and reestablishing of the same ecosystem to the immune suppressive tumor microenvironment. We confirm it also by histology. You can see here the massive infiltration of CD8, they lose their PD-1, but over time we have a reduction of those, a little bit of those cells, and they again re-express the PD-1 indicating for a suppression activity. But CD8 were not the only subset that actually were changed. And you can see here, this is an example of a different population that we like very much because they also express PD-L1, and they seems to eliminate almost totally, and they were co-associated, elimination of B cells was associated with the elimination of the exhausted cells. And furthermore, when over time, this population was accumulated together with the accumulation of the exhausted cells. So we further did a lot of characterization with the different markers, including macrophage markers, suppressive MHC, F480, name it, and basically we came into the conclusion that this population all of this population express CSF1R, and there seems to be two major subsets. One of them is the M2-like macrophages, and the second one is more of the monocytic dendritic-like. And when we sorted them, we asked whether those cells have the ability to suppress T cells proliferation. And here you can see that the reduction of those cells upon the treatment, but here you can see that their ability to suppress T cells proliferation. So the question is, how does trametinib actually affect those CSF1R positive cells? And it was intuitive for me to ask Manu, the students, let's see what is the level of CSF1 expressed by the tumor cells. So it was quite exciting to discover that trametinib actually reduces the level of transcript level of CSF1 in the cells. However, when you treat the cells for a longer time, here it was for almost 3 weeks, you saw that trametinib actually did not affect the level of CSF1. So that gave us the motivation to ask, okay, whether overexpression of CSF1 by the tumor cells can induce the same phenomena. So we took the 4QL, we overexpress it either with the GFP or CSF1, and we look for the efficacy of trametinib. So while in the GFP you had a stable disease, but then tumor progression, in the overexpressing cells, the progression was quite immediately upon trametinib treatment, and that was reflected also with the level of CD8 and CD11C. Furthermore, this overexpression of CSF1 was sufficient to prevent the tumor elimination induced by the therapy combination. So here you can see the tumor elimination of the treatment combination of trametinib and anti-PD-1 in 4QL and 4QT. But in the overexpressing model, this combination was much less potent and did not induce tumor shrinkage in any of those mice. This ratio between CSF and CD8 activation gave us the motivation to contact the partners, colleagues from Lyon, Pierre Santini, and basically he did a sequencing, transcriptomic sequencing on a cohort of 100 patients treated with either PD-1 and PD-L1, and we asked a simple question, whether the ratio between CD8 and CSF1 can predict or can indicate for a response, or in other way around, we can say what is the clinical benefit of treated patient with anti-PD-1 based on the ratio of CD8 and CSF1. And we have been able to see that in patients that they have higher level of CD8, that the ratio is high between CD8 and CSF1, they seems to have better clinical benefit compared to patients that they have high CSF and low level of CD8. So the take-home message from this study is that it's important to understand the dynamic of response. And basically, if you treat the patient with anti-PD-1, in our case, it seems not to work because the tumors are quite resistant to PD-1, but they are sensitive to trametinib. But when you give the treatment, you rewire the signaling machinery in tumor cells that reduces CSF1. I won't show you here, but it's via STAT, by the way. And basically, this time point of the stable disease, you have a massive infiltration of T cells. And in that time point, when you further activate the immune system, it seems to be to induce tumor elimination. However, if you miss the window of the treatment and the tumors start to reestablish the immune suppression ecosystem, supplementation of anti-PD-1 seems not to work that much. In the lab, we had a a lot of debate about the involvement of RAS pathway. And basically here I jumped into the fourth the story. And basically we look on the TCGA data and we saw that HRAS seems to be potent, seems to be a player, but Ofra Sankar was quite arguing about what is actually the role of RAS genes in head and neck cancer. So after this debate, they decided that they are going to sit and dig the literature and they export the raw data of hundreds of papers. And eventually they analyze 8,500 patients that have been sequenced for RAS genes. And those are the following outcomes. First of all, they show that specifically HRAS seems to be more dominant in South Asia. Moreover, they profile the mutation, the amino acid substitution in each gene. And basically, you can see that HRAS is more dominant, which is 61, following by the 12 and 13. KRAS is more dominant with the 12, and NRAS is with 61 and 12 substitution. Moreover, they had insufficient amount of N to look for what type of substitution. And it seems that with the HRAS, it's mainly the 12 goes from G to S, and the 13 is going to R, as well as in the 61 to R. And with the KRAS, we are more likely in head and neck having the D, which is, we are hoping to get the drug that the G12D inhibitor to try to exploit. There was another few pieces of critical information that was there somewhere, but now I think it's with this meta-analysis, it's more concrete. We have been able to show that HRAS is more dominant in the salivary gland and in the oral cavity of head and neck patients, while KRAS seems to have a trend in the sinusal cavity, and NRAS is slightly more dominant in the nasopharynx. This meta-analysis also showed that HRAS mutation is associated with the grade status, and that would link me to the last story that we are going to talk today, is the role of mutated HRAS in the oral cancer progression and metastasis. And this project is led by postdoc Sankar in my lab. And basically we ask a very simple question. How mutation in RAS in head and neck cancer can, and if at all, can induce metastasis? So the first experiment that it was, give us the first indication, we took a CAL33, which is all head and neck cancer cell line, and we overexpress the HRAS mutant V12. And we have been able to see that although those cells seems not to proliferate much in culture, their ability to migrate and to invade in vitro seems to be more active. Moreover, when we injected those cells into LSG mice, it seems to be more aggressive. You can see here that they grow faster compared to the HRAS script, and the number of enlarged lymph node and micrometastases in those mice seems to be higher compared to the CAL33 cells. When we analyze those cells in different approaches, we have been able to show that that HRAS induces the activation or overexpression of AXL and YAP. And by testing two different cell lines, two different PDXs, we have been able to show a similar trend. Here you can see H31 cells and SEC417B, and you can see that these cell lines have a higher expression of AXL and YAP, and that was associated with enhanced metastasis when you compare both. And this is also shown quite in a nice trend also in the two HRAS mutated PDXs gave us kindly by Jennifer Gravis. So we ask the next question, actually, is it true that HRAS can regulate AXL and YAP? So we did a siRNA for we knocked down HRAS and be able to see a reduction also of AXL, and when we knocked down of YAP, we also saw a slight reduction of AXL, and you can see that this reduction of AXL was associated with reduction of the ability of the cells to migrate in vitro. In vivo, we did a knockdown, stable knockdown of AXL, And you can see here the degree of knockdown. There's one clone that had a greater knockdown compared to the other one. But anyway, those two knockdown cells seems to have a reduction ability in their migration and invasion potential. And also in vivo, although that the cells seems to have some slightly more tumor growth proliferation advantage in vivo, their ability to form lymph node metastases and as well as lung metastases were reduced. We strengthened this phenomena by using an RTK inhibitor, the R428, which inhibits the AXL, the kinase activity of AXL. And you can see that the R428 treated cells showed a reduction in their invasion migration in vitro, but also in vivo. You can see that R428, at least in one model, for some degree, even enhanced the proliferation and the tumor progression, but the migration and the ability of the cells to home into the lymph node seems to be reduced. You can see here the reduction of the enlarged lymph node in the two models, in the H31, it's mutated, HRAS mutated, and the PDX2, which is also HRAS mutated. On the contrary, when we overexpress AXL, we have been able to enhance the migration. You can see here, this is the SEC17B that has a lower level of AXL, and the migration and invasion potential as well as metastasis in vivo is limited. When we overexpress of AXL, it seems like AXL is sufficient to enhance their cells' ability to migrate in vitro as well as in vivo, and you can see very massive macrometastasis in the lung, as well as increase of the number of enlarged lymph nodes in the tumor-bearing mice overexpressing AXL. We further strengthen this phenomena by using a model that we developed in the lab with the HRAS mutated, and I will make it short by saying that this is a very aggressive murine model that grow in NSG as well as in wild type mice, and you can see that the cells are very highly metastatic in NSG as well as but less in the wild type. Same observation seems like when we knock down HRAS and YAP, we have a reduction of AXL, reduction of invasion and migration in both models, as well as in vivo. When we knock down AXL, we've been able to show that although there is no significant differences in the primary tumor growth, the ability of cells to home into the metastatic as well as into the lymph node is reduced. And the question is whether AXL plays only a role in the migration and invasion, or it seems to have like more different roles, including in the lymphatic and the angiogenesis in the tumor. So we analyzed the tumors by staining of CD31 and LYVE, and we've been able to see that in the G-controlled tumors, we have more staining of the lymphatic system, while the SH AXL tumor seems to have less angiogenesis. When we took the conditioned media of those AXL knockdown compared to SG control, we saw that the conditioned media of the AXL knockout seems to have lower ability to induce tube formation. And here you can see the tube formation from G-control conditioned media, and the conditioned media of the AXL reduced that. That was also true in the human models. The two models that knocked down, the conditioned media have not been able, the UVEC cells to form tube, and that was co-associated with the reduction of VEGF-A and VEGF cells, VEGF-A and VEGF-C expression in the AXL knockdown cells. So far we learned that AXL, few more minutes, that AXL play a role in invasion and migration, induces upregulation of VEGF-A and VEGF-C that enables the cells to enable lymphatic and angiogenesis and host and metastasis. So the last part is to ask whether blocking the R428 blocking HRAS using TP ferritinib is sufficient to reduce metastasis and angiogenesis. And basically TP is a HRAS inhibitor, and it seems to be very active in HRAS mutated. And similarly also here, you can see that the TP treatment reduces the ability of the cells to migrate. TP treatment reduces the ability of the cells to secrete VEGF-A and VEGF-C, and TP also reduces AXL and YAP expression. Lastly, when we give the treatment with in vivo treatment with TP, we've been able to reduce tumor proliferation, reduces metastasis, as well as lymphatic and metastasis. So overall, I will summarize because the time is short. Summarize is that HRAS mutated are associated with advanced stage of metastasis. Mutated HRAS can activate YAP and AXL to promote metastasis. AXL is required for metastasis in HRAS mutated amplification. Mechanistically, AXL is involved in the expression also of VEGF and VEGF-A and C to induce lymphatic angiogenesis. When you treat the cells with a TP, you can reduce this phenomena, reduces AXL and YAP and VEGF, and basically to get a better response, you also need to inhibit the PI3K pathway. So I will summarize, just say thank you for all my students. Some of them are about to graduate soon. I will be very sad when they will leave. And some of them, like Manu, that now is a postdoc at NSKCC, and all the funding and my great collaborators that support me emotionally on crisis. Thank you very much.
Great. Thank you so much, Moshe. I wish you were here again in person to hear the applause and the congratulations to you and your team for really a beautiful body of work to bash out these pathways very methodically. You have a number of questions, so I'll get right to those. First question is from Shangjian Pan. Besides AXL, could you please elaborate other mechanisms of resistance to PI3 kinase inhibitors, and how about IGF-1 in particular?
So the question is about IGF-1R, whether it's a mechanism of resistance in?
Yeah, they're asking, besides AXL, do you know about other mechanisms of resistance to the PIK3CA inhibitors?
So the question is, the answer is, I'm sure that there are plenty. Okay, and basically, when we do the experiment, we trace after the one that pops the higher. But I would guess that if I will go deep into the data that we have been collected, I would assume that there are multiple mechanisms. And but it seems like AXL potentially can play a very important role, but there are other work by Jennifer as well as other that shows that also the ERBB2 and ERBB3 receptors play a crucial role in the efficacy, primarily in the HPV positive in terms of innate resistance.
Thank you. Another question. Is AXL expressed exclusively in the tumor cells, or have you detected it in the stroma as well?
AXL is totally not exclusive in the tumor cells. It's actually expressed quite high also on fibroblasts and in myeloid cells that have some suppression activity. So although that we are claiming by the in vitro studies that the combination is potent because we inhibit the receptor in the tumor cells, I think that in the in vivo scenario, there is a potential that inhibition of AXL has more of a general effect on the fibroblasts as well as the myeloid compartment.
Great, thank you. Another question from, I'm sorry, I don't know if it's Raul Aurora. How does the HPV status affect response to treatment with the RAS inhibitors?
On the RAS inhibitors, okay.
Yeah.
Not yet, I'm not there yet. I can tell you that we are about to chase this question, but we can do it so far only with the murine models.
Mhm.
To the best of my knowledge, there is no HRAS mutated HPV positive cell line, so we are not there to do so. Although that one of the PDXs can be HPV positive, I cannot. I'm sure. Okay, I can say based on different pathways that people, other investigators look on the mechanism of resistance, HPV positive and HPV negative, it seems that there is a potential role of the viral oncoprotein in the expression of the receptors. And this has been shown also by Grandis that E6 and E7 can attenuate it and regulate the expression of HER2 and HER3. So needs to be investigated, but this is something that I'm not sure that's been explored yet.
Great. And then a question from Jonathan Frieden. How do you think these will hold up, these signaling pathways and these molecular mechanisms, maybe pan-cancer, or do you think there's something unique to head and neck?
It's a tough question. So because I think there are some potentially uniqueness in some diseases, but I think the general molecular pathway in different mutations in the tumor cells can affect the signal rewiring. So potentially we can inhibit, I don't know, the EGFR, the MEP pathway in colon, in the head and neck, but the presence of KRAS mutation versus other mutation in the head and neck can also affect. So I think in general, I think that there are some general pathways that can be common between the different cancers, but potentially there are some uniqueness of response, primarily potentially in the HPV status, which is more unique for the HPV positive head and neck cancers.
Great. Another question, it says from Dagan Segal, beautiful work. Do you think AXL is involved in tumor growth?
It's not, I think, it's known that AXL is involved in tumor growth. It's involved in more invasion, and it's been shown by many investigators. And actually, there are a few clinical trials using either inhibitors of the AXL or conjugated antibody that can target the AXL expressing cells. So it seems to play a crucial role. And so my gut feeling is that it is.
Mm-hmm. Thank you. And I have a question. It's perhaps a little broad, but I'm thinking, your work to really bash out the timing of therapy seems very important. And, you identified a window of opportunity in the preclinical model. I'm wondering, what your thoughts are about moving that, clinically. Would this imply a neoadjuvant type of regimen or an adjuvant type of regimen? And maybe to tack on a second question along those lines, do you know anything about the metastatic microenvironment, if remodeling is occurring there as well in response to therapy, or if that window may be different?
I can tell you that we know very little. This is the major answer, but I can tell some speculations. I think that there is a good chance that the different lesions that present in different places in the body, they will respond differently, because also the availability of the drug, the pharmacokinetic, pharmacodynamic is different. And also the tumor ecosystem in the metastatic in the primary can be different between the liver, lung, or other organs. So I'm not sure that we'll have a general, but I would think that also with the immunotherapy, we can expect to have the abscopal effects, meaning that if there is a good response in one lesion that can potentially induces the neoantigen, the T cells activation, and immune memory that can migrate to the other region. So I'm not sure that in different places we can get the same trend, but it doesn't mean that the systemic effect won't occur. In terms of your question about the clinical trial, so I know that in France they are planning to conduct a clinical trial testing that. And in terms of window, therapeutic window in patients, I think it will require intensive study, or at least, in few patients to understand what is the dynamics of the response. And I think it will be fascinating to try to model it, I think, with either with the single cell sequencing or other approaches to understand the dynamic in patients. We can do it very nicely in mice, and we do it in mice, but I think in patients it could be, it can give more of a applicative flavor and crucial information for a decision of when and how to give and which immunotherapy we should give.
Mm-hmm. Yeah, great. I was asking you to speculate, so thank you. I mean, I think you took it more to reality, which is great. So along those lines, did you ever try a lead-in with immunotherapy first, and then I can't say it properly. Trametinib.
2 months before we published this paper, there was a different paper that took the same drug but flipped the sequential. They started with the immunotherapy and then added targeted therapy, a specific trametinib, and they also showed there that there is an additive effect. I cannot compare the two experiments, the two studies, but our study, we get tumor elimination, while in the other approach, there was more of a stable disease. The only few models had a tumor shrinkage. So to be fair, I would say that only if I will do the same experiments in the same lab with the same students, I will be able to have a fair comparison. But we did not explore that. And I think that this is something that the company that wants to actually develop the trial should put maybe more attention into, vice versa, if immunotherapy and then targeted therapy.
Nice. I think we have time for one last question, and it's quite an interesting question and relevant. So Jonathan Frieden's asking, he's an adult, and he's asked his PCP doctor to describe the HPV vaccine, thinking it might help him, but his doctor has refused. Is this something he should continue to pursue, in your opinion?
So the question is my personal opinion about HPV vaccination.
Yeah, HPV vaccination in adults, for thinking this may help in.
There is a fascinating JCO paper that actually quantified the percentage of the chance to develop cancer. It seems like we have been able to, we probably exposed to different HPV variants, and the people that are in high risk are those that actually seems to have a very active sexual activity with the different partners. So I don't remember the name, but you can, if you are in the group that that having a lot of partners, so more likely that it will potentially help you. But if it's you are in a monogamic relationship, I don't think that it will give any beneficial activity. This is just, again, speculation based on the results, and I give it, you know, the flavor of the natural life.
Well, thank you. Unfortunately, our time is up. This was a fantastic seminar. And again, I wish you were here to hear the applause you would be getting right now. Sorry?
I will come.
Yes. 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 I please do. I hope that time is soon. So thank you again, Dr. Alkabetz, for accepting our invitation and giving us a phenomenal seminar. And please congratulate your lab group as well on the great efforts.
Thank you very, very much.